Good evening, everyone. It is my pleasure to introduce the speaker of today's bio seminar, the last seminar of this academic year. And this is Dr. Nandini Rajamani. She is currently the assistant professor in the Department of Biology at IISER Tirupati. Now, we are all familiar with terms like climate change, and those of us with any interest in biology would also be familiar with how change in our environment is likely to have drastic effects on living things. A lot of Dr. Nandini Rajamani's research is focused on understanding the ecology of organisms and their adaptations with respect to changes in their environment. Her work, I feel, helps fill a big gap in understanding how climate change is likely to affect organisms, especially in Indian ecosystems. She does this with a range of approaches, including field research and using citizen science data. I'm in awe of how Nandini seamlessly contributes both to basic science and also engaging various stakeholders for the conservation of biodiversity in India. So, for example, she's involved in several initiatives to promote and communicate science to students, researchers, administrators, wider public. Also, since given where we are situated in Krea, also a greater focus on increasing focus on Eastern Ghats and biodiversity there. And I'm certain today's talk, featuring what I think are some of the cutest mammals, will be inspiring to many of us, especially those of us involved with the evolution course currently running at Krea. So with that, please join me in welcoming Dr. Nandini Rajamani. Thank you so much for joining us.
Thanks, Shivani, for that really nice introduction. I don't think anything that you said is really true, except for these animals being very cute. So that I think is undeniable, but I'm not sure about the rest. But thank you, and I'm happy to be here. Thank you for the invitation to talk about some of the work we do in the lab. All right, so I'm going to talk about some of the work we do in the lab, and I'm going to take you through a little bit of work from different systems. We do study climate change, or we hope to get to the point where we can talk about how species respond to climate change, both in the Himalayas as well as in the Indian subcontinent. I might not be really talking about that work today as much, but basically, I think I might prefer to talk about how we understand adaptations of animals or small mammals in the first place. So as we all know, we live in a changing world, and we know that India is poised for pretty severe climate change in the next few decades. And species respond to all kinds of changes around them, including changes in climate. But even within one species, there's a lot of variability. So the poster child for climate change, the polar bear, is also such an example. If you look at the pole, that diagram over there shows you different populations of bears that are responding in different ways. Some are actually increasing in number, some are decreasing, some are constant, and some we don't know anything about. So even for one species and one threat, the response might not be consistent. And this is what makes it exciting to study species like this. Species are constantly adapting to conditions around them. And you might know this. This is the example of the finches from the Galapagos. The Grants, Peter Grant and Rosemary Grant, have studied these finches for about 40 years now. And we know astonishing things about them. What's most exciting is that we see evolution or adaptation happening in really short periods of time. We were taught, at least some of us, that evolution is this long drawn out process that you don't really get to witness in one human lifetime. But we've seen more and more examples of adaptations and possibly evolution happening across systems. This is one excellent example where there was a drought in 1977, and the repercussions of that drought on both the plants and the birds have caused a shift in beak size. So yes, beak size is the trait of interest that was measured by the scientists in these studies, but it's exciting to see how one incident or one event has really had has caused a complete shift in population traits. So this is an example of some kind of selection at work, something we can measure and see in a matter of decades. We also typically see species responding to altitudinal change. We've seen this in many small mammals. We see examples of historic range shifts that happen in present time. Species that live at different altitudes might respond differently. This is a pika from the Trans-Himalayas. Again, pikas are a poster child for climate change. Studies on the American pika have told us that with global warming and climate change, species lose habitats when they are restricted to high altitude systems. So yes, the American pika has kind of been range restricted or has moved up mountains, making it a species of conservation concern. So in my lab, we try to look at species adaptations, and we look at how species respond to changing conditions around them. And while we can say that we just talked about typical range shifts, species low elevation species move up, high elevation species lose their ranges. There's also other kinds of changes that happen when external factors like climate or habitat change. And this is an example of a behavioral change. This is on the left here is your life cycle for a marmot. Marmots hibernate for about 6 months in a year, and that's the gray part of the cycle there. And they're active for about 6 months in a year, and this varies. It's a 4 to 6 month window of active period, depending on the species and where they occur. So what happens with climate change? So in this 6 month window, in a regular part of their life cycle, marmots emerge, they mate, they give birth, the pups then emerge, and all of these marmots have to put on weight before they go down to hibernation again. So what happens with climate change? It's kind of contentious, and data from different sites tells us different things. You would expect that the activity window might be longer, and that this is a good thing, but it's possibly not. And studies have shown us that short-term effects versus long-term effects can be very different. And it's important to study some of these populations over long periods of time to really understand what's going on. So in some sense, in my lab, we look at populations, but in order to study a trait or to pick a trait and look at what impacts that trait or causes that trait to evolve in response to something, we need to know what traits are under selection or why traits have evolved. And we can do this in different ways. So today my talk is going to focus a little bit on the evolution of traits, or it's going to focus a little more on the evolution of traits, right? Though we will intersperse climate and environment through the talk. All right, so what taxa do we study? In my lab, we study squirrels for the most part. We focus on palm squirrels and marmots. We also study giant squirrels. We study non-squirrels. We call ourselves a silent lab. We study pikas and some small carnivores with collaborators. We work in two landscapes primarily, the Trans-Himalayas, and it used to be the Western Ghats, but work is now slowly spreading across the Indian subcontinent to the Eastern Ghats as well. How do we do this? With a number of tools and methods, like Shivani mentioned. And our focus is right now in the lab are looking at evolution of traits. So we want to know which traits are adaptations and which traits are simply evolutionary relics, right? They've just been dragged along the phylogenetic tree. Some traits are under active selection or provide certain advantages to species. We also look at species that live in specific environments and how these environments shape them. We talk about habitat association. I'll talk very little about this today, and maybe we can discuss some of this at some point of time. All right, so we work or we look at two time scales. We look at evolutionary time and we look at what happens in current time. We use different methods to do this. With evolutionary time, we use comparative phylogenetics or genomics. We also use a lot of modeling. And to study changes in current time, we use behavior or natural history or experimental studies. Like I was saying, we work in two landscapes, the Trans-Himalayan landscape, mostly in Ladakh and Kashmir. And we work in southern India. And this is where we work on the palm squirrel system. So I'm going to talk to you a little bit about the work that we do in both of these landscapes, a little bit about marmots and pikas, and then we'll come down to the south. So in the Himalayas or the Trans-Himalayas, so just for people who maybe are not aware, the Trans-Himalayas are the highest altitude plateaus and rolling hills in the Himalayas, and typically areas above 3,000 to 3,500 are called the Trans-Himalayan landscape. A lot of where we work is around 4,000 meters above mean sea level. We work in these meadows, these rolling open meadows, and we have small mammals that can be marmots, can be pikas, and can be voles. And right now in the lab, we only study marmots and pikas. So they live in these really extreme environments where the temperatures can go down to -40 in the winter. And in the summer, it can go up to 35. For one brief month, it can be quite hot. Most of the time, though, it's not 35, it's quite cold. And so how do species adapt to such extreme environments? When we look at two species that can live in the exact same meadows, literally running around each other, sometimes sharing burrows, do they have the same adaptations to live in these landscapes? And the answer is no. Mammoths and pikas have extremely different adaptations to the same exact microhabitats and climatic features, right? So mammoths, like I was saying, are hibernators. There are about 15 species of mammoths globally. Mammoths are squirrels. Mammoths can be really large. They range from about 6 kilos all the way to about 12, 13 kilos in the case of some species. So they look like big dogs, and they're hibernators in the sense that they sleep or are hibernating for about 6 months in a year and are active for 6 months. So all of their annual life cycle is compressed into this 6-month period. And so time, the habitat, and the resources are extremely crucial for them. They deal with the extreme cold by not dealing with it. So what about pikas? Pikas don't hibernate. Pikas are small. They're about 100 grams to 300 grams or 400 grams. There are several species of pikas. Pikas can live in the exact same meadows as marmot and eat the exact same food. Sometimes they have the same predators. Pikas don't hibernate. They are out all winter, and they deal with the cold in very different ways. So we started out trying to see what these differences were and what caused these species to live like this. Very little was known about marmots and pikas when we started our work in 2017, and this is especially in the Ladakh landscape. So the first thing we did was a survey for all of these species. Now, marmots are large and visible and quite easy to spot once you get the hang of it. And we could do a lot of surveys and visual counts to look for where colonies were dispersed. So here I'll talk to you a little bit about the marmot work. I'm not really talking about the behavior work that we've done, very little about the genetics. All right, so a lot of this work was led by other researchers in the lab. This is work that Senin has led, though there was a team of people studying marmots. So when we started our work in 2017, we knew very little about where marmots occurred. There are two species that occur in India, the long-tailed marmot and the Himalayan marmot. If you look at this map here, the long-tailed marmot's distribution kind of stops, doesn't extend into Ladakh, not even into Kargil, according to the IUCN map. While the Himalayan marmot has this ring-like distribution around the Tibetan plateau. And we did surveys. All these round dots are our data points, and the squares are secondary data points. We found surprisingly that most of our sightings of the long-tailed marmot were outside of what the IUCN says for the Indian range of the long-tailed marmot. We really know very little about these species, even though they're so large and visible and quite easy to spot. So, all right, so now we know where they are. And is this habitat different? You can see that there doesn't seem to be an overlap between the two species. We found the two species within 15 kilometers of each other, and we suspect there might be valleys in the Zanskar range where they could be closer. We've never seen them in Simpatri though. So when we did some kind of modeling to try and predict what environmental variables determine the occurrence of both species, we found that the long-tailed marmot occurs where winters have intermediate snow. We found very high correlates with ice and glaciers for the long-tailed marmot. We did not see this pattern for the Himalayan marmot, which was quite surprising. So just to summarize, the Himalayan marmot occurs at a higher altitude than the long-tailed marmot. The long-tailed marmot's elevation range ends at about 3,500 meters. The Himalayan mammoths range starts about there, though you have species crossing this boundary, not in sympatry though. So the Himalayan mammoth goes all the way up to 5,700, 5,500 meters, very high altitude species. So we assume that this would be the species that occurs in the colder regions, the Trans-Himalayan Desert, extremely dry, extremely cold. To our surprise, we seem to think that, or an analysis seems to suggest that the long-tailed marmot possibly inhabits areas with a different kind of cold. And this cold is linked to snowfall, which is not what we saw for the Himalayan. We saw that the Himalayan marmot is probably limited by high summer temperatures, and this is probably one of the reasons it's not found on the Tibetan plateau. It's probably too hot for the species on the plateau, right? And this is summer temperatures. And when I say too hot, I mean a maximum of 20. Ladakh has points and periods and locations where you have temperatures going higher above, but this is through modeling and remotely sensed temperature variables. So basically, there are two species here that are extremely cold adapted species, but seem to be different, maybe a slightly more wet adaptation to cold and snow for one compared to the other. We looked at some genetic structure for populations, and I'm not going in depth with the marmot work right now. I'm trying to save the depth for the barn squirrel work, which is in the second half of the talk. But just to say that we've done very little genetic work with the two species, but we already seem to see patterns. So the long-tailed marmot is the lower elevation species, but there's not so much of low elevation habitat that's available. They are threatened both by climate change as well as grazing. So long-tailed marmots seem to be restricted to certain mountain tops along some of the ranges in the Himalaya. Where they are located in such isolated places, it looks like their populations seem to be genetically distinct. Again, this is very preliminary work, and maybe by the end of the year, we'll have a clearer picture about what's happening. The Himalayan marmot, on the other hand, doesn't seem to show any kind of genetic structure with population, and no rivers seem to form barriers and no valleys seem to form barriers. So this is one large interconnected population on the Indian side of the Tibetan plateau. We have not surveyed outside of India, so we don't know about its global distribution, but within India, it seems to be one large contiguous population. So now that we've talked about marmots, we'll move on to pikas. But with marmots, some of the other work we're doing is we're tracking colonies over time to look at emergence dates and elevation, and hopefully we'll be able to integrate this with a knowledge of climate. And we also want to look deeper to look at population dynamics, not just across space, but also within groups. They seem to have very distinct sizes, group sizes, and behaviors. All right, so we've done a lot more work on pikas than we have on marmots, simply because pikas are smaller, easier to access, and there was a PhD student who worked entirely on pikas. So I will talk to you a little more in detail about pikas compared to marmots. For those who don't know, pikas are not squirrels. They are in the rabbit family, and the lagomorph family, or the family that the rabbits come from, are divided into three main kinds of mammals. You have pikas, which are small, live in cold mountains, usually high in elevation. They can live in groups, or they can live alone. What's of interest to us is how they live. Do they burrow under the ground, or do they live in rocks? And they have young that need care. So they have systems that enable the female to look after the young. With rabbits, they're larger than pikas, medium body sized, widely distributed, and again can be social or solitary, and very similar in terms of altricial young needing care. Hares are the largest in the family, and their habits of burrowing or living under rocks varies a little across species. So when we looked at pikas, most pikas, there are 30 species globally, are distributed in Asia and North America. Many of them live in very high elevation conditions, higher than 4,000 meters, and these can be extremely hypoxic places with low oxygen. Pikas don't hibernate over the winter. Instead, they collect and store hay, and they do suffer high winter mortality as a result. So with pikas, our interest is within pikas, there seem to be two life history strategies that pikas employ, or lifestyles, right? So one is what we call a rock dwelling lifestyle, and one is called a burrowing lifestyle. So these are just two examples. The American pika, which is really well studied, is an example of a rock dweller. And the plateau pika, which is something that occurs in Tibet, also fairly well studied. What do we know about them? Litter size can be variable, much larger for the plateau pika. The plateau pika is the slightly larger animals. They mature early, and they are short-lived animals. Densities can be extremely high, 300 animals per hectare, and they are social animals. What's interesting is that rock-dwelling animals are asocial animals, long-lived animals, low-density animals. So if you look at these contrasts, and these are just examples of two of these lifestyles, you can see that their life history variables or characteristics seem to be different with respect or according to whether they burrow or live in rocks. So the big question for us was, is this really a strategy to burrow versus live in a rock? Does it really determine many aspects of your biology or behavior? And so we wanted to look at why burrowing evolved, right? And so bear with me, this can get a little convoluted, but we looked at several hypotheses, some of which were mentioned in this slide before. One of the associations was that burrowing species are more fecund, and our predictions were that burrowing species have larger litter sizes, and they have more litters through their lifetime. So there are also probably associations of life history with morphology. Maybe small size species are more fecund. Social species should have more young and be able to breed faster and more litters per year. Or there could be associations with ecology, where life history is associated with thermal regimes, right? And our predictions were that at higher latitude, there is less optimal time for breeding. Therefore, it makes sense to have larger litters and put all your kind of effort into one breeding episode. So we created a phylogenetic tree and mapped burrowing across the lagomorph tree. This includes rabbits, hares, and pikas, and some outgroup rodents. The white dots are your burrowing species, and the red dots are non-burrowing species. And we see that burrowing has evolved multiple times in Lagomorpha. So our theory that burrowing evolved with hypoxia or high altitude was not something that we found. We also found that the ancestor of all Ocotonids, or the entire family, is a species that lived in the burrowing state, right? I'm sorry, of pikas is a species that lived in the burrowing state. Of all hares, the ancestor of all hares probably lived in a non-burrowing state. So if you want to look at transitions of non-burrowing to burrowing behavior, this seems to have happened 11 times, only twice in pikas. And this was completely contrary to what we had expected. So with pikas, we find that rock dwelling to burrowing has happened only 2 times, but a switch from burrowing to rock dwelling has happened many more times. So this seems to suggest that all pikas were burrowing species, and at certain points of time, they lost burrowing, they lost sociality, they probably evolved very distinct asocial, low breeding, slower rock dwelling life history strategies. Why? Well, that's something we're still trying to figure out. So in order to look at why, we did some analyses with environmental variables, and we found that we found a correlation with latitude, which automatically implies there's something to do with temperature. So a lot of our temperature variables were significant. So we think that burrowing does confer thermoregulatory advantages in cold climates, not just the act of living in a burrow, but also possibly in a social manner. Multiple animals sharing burrows, or having independent burrows that lead to a large connected burrow at some point. We did not find patterns with sociality or body size or elevation or any of those variables. With how many young animals have association with offspring size, we found that contrary to what we expected, larger species seem to have more young. So if you're familiar with traditional life history theory and R and K strategies for species, and this is the mouse to elephant kind of idea. The idea is that smaller animals like mice reproduce faster, have more young, live fast and die young, right? But you have something like the elephant, which has very few offspring per litter, one usually for an elephant or two. And these litters are really spaced apart across the lifespan of this animal, right? So this is your traditional explanation of the life history continuum. And we did not see this with pikas, surprisingly. We saw that larger pikas have higher litters and more frequent litters. We still are not quite sure why this is, but it might be some other advantage that thermal regimes and life history or lifestyle strategies confer on them. So while it's great to do phylogenetic analyses and evolutionary analyses across a family, this still does not tell you why individual or what benefits individual species derive from specific behaviors like burrowing or rock dwelling. And you could ask, well, how do you say that rock dwelling provides a thermoregulatory advantage that is lower compared to a burrow? Yes, of course, it's a very valid question. And we attempted to study this in the field in Ladakh. So we were lucky enough to find locations where we had species, or 3 different species that lived close by within a span of a few kilometers. So we assume that the temperature conditions in those few kilometers is not going to change too much. And then we used eye buttons or temperature loggers, found refuges, which could be burrows for the Ladakh pika or burrows for the Nubra pika, or rocks for the larger pika. So if you can imagine, we're working at altitudes of 4,000 to 4,500, all the way up to 5,000 and above, and walking around manually trying to find these refuges. So for the social species, Ladakh pika and Nubra pika, you find a colony, and then you stay put and count your animals and walk around. But for something like the rock-dwelling pika, it's incredibly difficult. It's simply a matter of luck to be able to see this animal. Most often you don't see this animal. You're simply walking around looking for fecal signs or other material around the rocks. So we did find enough refuges. And we tried to use these thermal buttons or temperature loggers to look to see if a refuge provided a buffer against external temperatures, right? So the idea is that a burrow is going to give a pika more of a thermoregulatory advantage than a rock. So the idea is just physics. Rocks will heat up when the temperature is warm. They take time to cool down. We know that different depths or sizes of rocks provide different advantages, but we assume that a burrow, which can go down deeper, will provide better advantage over any combination of rock. Right. So when we did some preliminary experiments and we left eye buttons in these burrows, both inside and outside in a paired experiment, we found that rock refuges are cooler than outside temperatures during the day and warmer than outside temperatures during the night. So rocks do provide significant thermoregulatory advantages. But then we looked at rocks versus burrows to see if burrows were better than rocks. Overall, no. But if you looked at the day, burrows seem to insulate temperature better in the day than rocks did. But at night, they both functioned in similar manner. We must say that we didn't do this work in the winter when it is probably critical to study thermoregulatory advantages. This was all work towards the end of the summer. It was starting to get cold, but not nearly as cold as the winter. And we were planning to go back in 2019, 2020 for a winter season, but COVID really derailed all of that. So we haven't been able to do the winter experiment yet. All right, so when we looked at 3 species, the when we separated the data not by burrowing and rock, but by species. We have two burrowing species, the yellow and the black or gray, and the green is the rock dweller. You can see there's high variability in a species like Ocotona nubrica, and that this variability decreases through the day. So there's a lot more that needs to be done before we can be absolutely sure of our results. And ideally, we need winter sampling before we can state conclusively whether burrows or rocks are more effective at buffering a species of animal from the temperature. All right, so I feel like we attacked this whole question of burrowing in different ways. We looked at a phylogenetic study. We did a little bit of behavioral analysis. We've done field experiments. We've done field measurements, and this is a paper on microhabitat selection that is just being submitted. We're also doing some genomics to look at regions of the genome that could indicate different behavioral traits, whether they be burrowing, which is very, very complex, very ambitious, or something like adaptation to cold, which is much better studied, so it's easier to get a handle on that. We also think there are other behavioral strategies that animals employ to deal with the cold. And this is a paper that should be out pretty soon. We observed pikas storing food as a strategy to overcome food shortages in the winter. This is a behavior called haying. Many species do it. The animal cuts and carries food back to its burrow or hollow, dries the food. It shouldn't be maypoles, it should be hay piles. I think I corrected autocorrect about 6 times, and I missed this one. All right, so they dry food in hay piles to last through the winter. And these are the first observations of any trans-Himalayan pikas haying. So haying would be pretty important because some of these hay piles can be really large. And we saw this behavior in both of our burrowing species. Pikas started to hay sometime around August, and individuals would simply have these piles that just grew and grew. You can see some outlines around these hay piles. This was early on, and by the time we finished fieldwork around the end of September, early October, some of these had become really large. When they dry, these animals take them down into the burrows for the winter, and you might not see hay piles in the winter because they're probably underground. Some reports of hay piles from other species have said that hay piles can be up to 8 kilos in weight, which is extraordinarily large, right? We didn't see anything this big. But what we did see was that there was variability within a species and there was variability across species. So the more cryptic Nubra pica made these really sporadic, disconnected hay piles that were really close to their burrows, very amorphous, no real shape or size, and not very large. The Ladakh pica, on the other hand, which lives in these open meadows, had huge hay piles that were defended by individuals. A large colony had many hay piles, different in size and structure from a smaller colony. So there's high variability, and animals clearly have behavioral strategies to deal with the cold. Ideally, we need to follow them again through the winter to see how behavioral strategies like food-related strategies help them survive the cold. So I can take questions about the Ladakh system now, if people have questions, or I can just continue and we can keep going. But that's one of the colonies that we worked on. But it's incredible. This is at 4,600 meters. It's brutal, but it's totally worth it. Yeah. All right. So if there are no questions, we'll move on to talking about squirrels. And we'll talk about something the lab has been working on for about 4 years. We are working quite extensively on squirrel color. And everyone knows what squirrels are in India. Squirrels are really common. They're part of human history, human mythology. In India, people have noticed squirrels for over 2,000 years. We have reports in Tamil Sangam poetry of squirrels from 300 BC, right? Or something like it. There's a bit of a window with dating. this kind of portrait. Despite the high diversity of squirrels and how common squirrels are not well studied. Even something as common as the palm squirrel that is literally in everybody's backyard in the Indian subcontinent. So we started to study palm squirrels simply because they are everywhere. We really don't know too much about them. But I'll talk about one aspect of what we do with palm squirrels. Other things we do with palm squirrels are distribution. We look at adaptation to changing environments. We look at responses to climate, possibly. Again, maybe a couple of papers will be out by the end of the year. But I'm going to talk to you about the evolution of color and patterns in palm squirrels. So we have 4 species of palm squirrels in India. We have the 5-striped palm squirrel, the 3-striped palm squirrel, the jungle striped palm squirrel, and the dusky striped palm squirrel. The dusky is the smallest species, and it's the most range restricted, found only in the western Ghats of India and in some parts of Sri Lanka. Very difficult to see, very cryptic, very shy animal. So for the most part of what we do, we're going to focus on the other 3. The other 3 are very different, all very common where they occur. They are all commensal in nature, do not shy away from humans, and possibly explode in numbers around humans. That's also something we're looking at, and I'm not talking about that work today. So if you look at this map, the 5-striped palm squirrel. So when we say that these are striped squirrels, you have to count the light stripes on their back. So if you look, there's like a dark saddle patch on the squirrel. So the squirrel is a shade of brown, and then there is usually a dark saddle on each of these animals, which is a darker shade of brown or almost black. And there are 3 or 5 light, pale cream to whitish stripes on the back. So these are the stripes. You don't count the black as the stripe, but you count the light parts as the stripe. Oh, and everybody must know the story. How did the squirrel get its stripes? It is, of course, because it served a god, and the god rubbed the squirrel and said, "Good job," or something. And, that's how the squirrel got its stripes. So given this really fascinating story, we said, okay, let's try and see if we know what these stripes actually do. Maybe we can actually answer the question of how these squirrels got their stripes. All right, so when we have the 5-striped squirrel, it's mostly Central India and North India is where it's distributed. There are records from the south, but this map seems to show that there seems to be some kind of limit. There is a large zone of overlap between the 3-striped palm squirrel and the 5-striped palm squirrel, but the 3-striped palm squirrel kind of stops around Central India. The jungle striped squirrel, or Funambulus tristriatus, is distributed along the west coast of India, clearly in the more wetter areas and along the Western Ghats. All right, so with squirrel pattern, like I was saying, if you look at a squirrel, you see color, you see different kinds of color, you see a base body color, which can vary. We're only talking about the dorsal color. The underside is usually cream for most animals, shades of cream. We're talking about animals that vary from a more grayish pelage to a really dark, rusty brown pelage, right? And then on the dorsal area, there are stripes, and these stripes can be 5 stripes for the 5-stripe or 3 stripes. They can be really dark, distinctive stripes like the 3-striped palm squirrel, or really pale, insignificant stripes like the jungle striped palm squirrel. So clearly there's a lot of variation just in this one genus and a few species in how the squirrels appear. So traditionally, we don't invoke sexual selection as a hypothesis for mammalian color for the most part, though there are exceptions, especially with primates. And the general idea is that color and pattern confer some kind of camouflage or predator avoidance. So our questions was, is this true? And why have both color and stripes evolved? And why do you see differences in color and differences in stripe patterns? Can they both be explained in the same way? So we started out by looking at museum specimens. We went to the BNHS museum a few years ago and collected, measured, photographed in constant light conditions, all of the skins that they had. And this is the kind of distribution of samples that we have, about 800 skins that we're using for analysis. So each of these skins is photographed in a light box, then the data is color corrected, and then we use softwares to extract data from these images. And we do this in different ways. There's pattern analysis as well as color analysis. So we started out looking at all areas of the body, the head, the snout, the forelimb, the hind limb, the rump, the tail, and then finally narrowed down our analysis to the dorsal area only, because that's where we see the highest variability. So all the results that I'm going to be talking to you about today have to do with the dorsal region only. And so the first thing we did was look to see if there's a geographic distribution of color of squirrels across our areas of sampling. And we see that overall, we see high correlates with latitude and longitude. So we see that darker colors across, if you pool all three species together, darker colors occur in the lower latitudes and in possibly humid environments, right? Lighter colors occur in lighter latitudes, basically in the Indian subcontinent. As you go north, you will see lighter colored squirrels. And this is typically predicted by theory. This is what we predict for Gloger's rule, as Gloger's rule, which is a rule that explains humidity and color evolution. We see a longitudinal gradient also, and this is more apparent for Palmarum and Pennanti, which are the more widely distributed species. We see them getting lighter as we go eastward, possibly as there's also like a drier element that's happening with longitude from west to east. For Tristriatus, the species that is along the west coast, we see a very strong latitudinal gradient, but no such longitudinal gradient. So we looked at environmental correlates, and we find that of all the variables that we examined about 20 variables with correlates with environment, with habitat, and with climate. And we find that rainfall gives us the strongest model. So we look at precipitation and different variables of precipitation. We see again the pattern to be the strongest with Tristriatus. So when we say effect, that's mean color. It's a principal component variable of color that's been extracted from multiple regions on the squirrel's body. And in case someone's interested, this is a composite of 3 channels of color: red, green, and blue. We did not find any one channel to overshadow the other 2. So it's fairly equally distributed across these 3. We see patterns with rainfall for all species, but it's strongest for the jungle striped barn squirrel. Also, we see average rainfall. There are 2 different variables of rainfall. Again, we see patterns with average rainfall. I think the one before was total precipitation, and then average. But again, the patterns are strongest for the jungle striped squirrel. Here we looked at 3 variables. We looked at total rainfall, we looked at average rainfall, and we looked at seasonality of rainfall. We find that at lower levels of precipitation, squirrels are dark. Even at lower levels of precipitation, squirrels can be dark when there is a higher average rainfall or high seasonality. So basically, seasonality seems to be really key. When seasonality of rainfall increases, squirrels get lighter. And this is a pattern for all species across India. And the pattern holds when we do species-wise analysis too. So the next step that we are taking this analysis to, and this is work that's still in progress, is trying to see what a predator sees. And this is really like taking color analysis to another level. First, what does a predator see? And how do you know what a predator sees? There are models of predator vision that are out there, and you can model what a bird sees versus a snake, versus a ferret or a cat, versus a human. And we use a software that has inbuilt libraries for vision. And when these vision libraries are not available, you can build your own and use that for analysis. So basically, when you put a photograph in, its default looks at the default that it gives you when you do an analysis of patterns is how a human views something. You can change the lens of the software to see what a cat sees or what a raptor sees. And just to kind of tell you, a raptor sees colors that we don't see. They see UV. They see birds see way more color than we do. We are trichromatic primates, see 3 color bands. Cats and many smaller mammals, many mammals, don't see as much color as we do. So ferrets and cats have very different vision systems compared to humans, right? So it's one thing to say that this is what a predator can and cannot see. It's another thing totally to apply a vision system to your analysis. So in this analysis, I'm not going too deep into this, but we're asking if pattern has evolved to confuse predators. And we looked specifically at the stripes, and stripes provide, it's theorized that stripes and spots provide a kind of disruptive signal, which doesn't allow a predator to see this animal very clearly. So does the edge, or do the stripes allow a breakup of the edge of the animal, right? So there are different kinds of analyses you can do. And this is one analysis called boundary strength analysis, which tells you whether this animal, whether it's a ferret or a raptor, can see the difference of the alternating stripes and the boundary changing from light to dark, or this transition well, or if it cannot see it well. We see differences with what a ferret can see and what a raptor can see, and it's largely species specific in terms of the predator. And the predator sees all 3 species of palm squirrels more or less the same way. So this is really interesting, but like I said, this is really preliminary analysis, and it should be done in a couple of months. But when we use multiple variables of metrics to look at, you can look at intensity of edge, you can look at boundary strength, you can look at absolute color that a predator sees, we started to see slightly conflicting results. So it's quite difficult to interpret some of these if you don't know. Sorry, did I just lose the talk? Yeah, all right. If you don't know exactly what you're looking for, right? So when we looked at color, we kind of pause here. We do see strong environmental correlates that color has evolved in response to, specifically rainfall. All of this follows very old established theory about color. We see the same trend across 3 species. And we see that the species in the most wet, humid regions shows this pattern in the most strong way. With respect to what predators see, we think different properties of stripes are differentially visible for predators. And this is what makes it really complex, right? So the last thing I'm going to talk to you about is a behavioral study where we take this analysis of color and what a predator can see, hopefully one step ahead. So we've been looking at behavior and whether an animal can modify its behavior based on where it is, and therefore either increase or decrease the camouflage efficiency that it has, right? So what if an animal has a certain kind of stripes or certain kinds of colors, does it know that its stripes will be more visible or less visible on certain backgrounds? And does it behave accordingly? So is there an added element of how this animal interacts with the environment that it lives in? So to do this, we followed squirrels in the field, recording behavior and substrate use. We did this in a semi-rural environment. The squirrel, we tested hypotheses about whether squirrels are cryptic in the background or whether they optimize camouflage through substrate selection, and whether it does different things on different substrates, and these different substrates provide different levels of crypsis. We did this work in Kodaikanal and in the villages around Kodaikanal. Again, this is a master student who's just submitted her thesis from IISER Tirupati, and she's defending next week. And we studied 2 things here. For every observation of an animal, we studied behavior, and then we studied also color and camouflage for that animal in that particular location or in that particular behavior. So we had a combination of photographs and videos for all animals, and videos were analyzed with a software. Color and pattern were analyzed with another software. So in short, where is a squirrel more cryptic? If we just divide the area into ground and non-ground, we see that the squirrel is more camouflaged on the ground compared to non-ground. And again, here we use 2 visual systems, a ferret system, which is a cat system, and a human system, which is a primate system. So both cats are major predators, bonnet macaques also eat squirrels, right? So we see that whatever metric you use, you come up with the same result, whether you're looking at luminance, which is simply a colorless estimate of brightness against the background, or you look at edge disruption of a striped animal against whatever background it's on. We say that the animal is more camouflaged on the ground compared to non-ground, right? So does it spend a lot of time on the ground? No. So squirrels spend much more time on non-ground surfaces, which is surprising because these surfaces don't give them as much of a camouflage advantage as the ground does, right? So what are they doing on these different surfaces? Squirrels seem to forage on ground surfaces where they have higher camouflage, and they forage less on non-ground surfaces. But when squirrels are resting, they seem to rest on non-ground surfaces where they have lower camouflage, right? So squirrels seem to differentiate activities that they perform on different surfaces. What if it's not simply a height-related issue? We also categorized all these surfaces into complex or non-complex backgrounds. And this we did after much discussion and consensus. And we find that, again, we find complex surfaces provide more of a disruptive advantage under both vision systems. Not significant for the ferret vision system, but when we look at humans, it's a statistically significant result, right? So when we divide surfaces into complex or non-complex, the left is an example of a complex background, lots of leaves and twigs. The right is more simple, so we categorize them relative to each other too. We see that in any metric, whatever metric we take, squirrels spend more time on non-complex substances, substrates, and they move to complex substrates really frequently. So in some sense, they reduce the continuous duration of time that they spend on a non-complex background, and they're constantly moving back and forth depending on what they're doing. So what are the behaviors that they perform in different substrates? When they feed, they are usually on complex substrates than on non-complex substrates, right? So this is really exciting for us because it seems to suggest that it's not simply color or pattern, but there's also behavior. Does a squirrel know where it's more visible to a predator? Possible. At least the squirrels we studied seem to feed on surfaces where they were more camouflaged, but they seem to be okay to rest on exposed surfaces. And this is maybe because they can respond to predators quite quickly. In fact, when they rest, they're usually looking down or looking around for predators and alarm calling as soon as they see something. So yeah, I mean, this is the kind of work we are doing in the lab. And I wanted to kind of talk a little bit about different aspects of work stop here. And I'm happy to answer any questions you might have. The first four names are the people whose work I have featured in this talk: Senin, Harsha, Niveta, and Harsha Jagdish, Harsha Kumar and Harsha Jagdish. But yeah, this is the lab. We have a website. We also have a citizen science program that Shivani was talking about. I didn't talk about that today, but we have two social media handles, one on Instagram, one on Facebook, and hopefully we will set up a website on Squirrels of India by the end of the year. So if you want to find us, look for Squirrels of India and you'll find our way back to us. Yeah. Thank you. Thanks a lot, Shivani.
Thank you so much. If there are any questions, please feel free to raise your hand. I can see some of you are sitting in a classroom. So anyone there, feel free to raise your hand here and we'll let you, I'll just call on you to ask your question. Nandini, may I request you to maybe stop screen sharing? So, thank you so much. Maybe while there are questions being formulated, I can ask one. So I was curious about the squirrel data that you collected from from BNHS and from other specimens. Did you see any, or did you look for any relationship with body size? And do you think that could play a role too in other differences that you see?
With color and body size? Yeah, we haven't looked, I mean, we haven't found any differences with body size. We also looked at gender. We looked to see if males and females show slight differences in coloration, and we found no patterns there either. But this is only the dorsal color. There might be something going on with the color on the head, which I suspect is a seasonal thing. We don't have enough data on this.
I see.
Like in some times of the year, you can see a lot of rusty orange color on the head of a squirrel. And that's something that we would love to study, but not enough data from museums.
Hi, Nandini. Nice to
Hi.
see you again.
Yeah, nice to see you too. Yeah.
So very, very nice talk.
Thank you.
Very nice talk. And I was really fascinated by all the kind of thing. And like Tibet, always a kind of a bucket list to do. And so, but so I also like involved in evolution course somehow with Shivani. So it was just when things were coming up, things were popping in my mind. And I have a like question wanted to ask you that, I mean, just like finches, so you said that these small mammals, those who live in the mountaintop. So have you been like far enough to see that if there is like difference or speciation within these in that different geographical places? Have you encountered anything like that or not?
That's a really cool question. Even with mammoths, we saw a lot of color variation across the landscape where we did surveys. We have genetic samples, but I doubt they are different species. They're not separated geographically. They're not isolated enough. And we strongly think the color of the marmot depends on the soil color. Again, we did some very, very crude analysis and found some patterns, but nothing that we can publish or really talk about.
Okay.
With one species that's distributed across Afghanistan, Pakistan, and India, when you look at photos, they look extremely different from each other, though they are classified as one species. There is probably something going on, either very high polymorphism, which is possible in squirrels and does happen in many, many species. But again, I am not in a position to say.
Okay.
Yeah,
So,
we
I
simply
mean,
don't know.
and so do they have like similar calls, like mating calls or help call? I don't know what different calls. Do they sound similar or like birds, they are like different?
The two species or within a species?
Well, like two species or within species, I mean, how different they are? They're very different. Can you like really tell by voice that it's
Yeah.
Yeah? Okay.
Yeah, the long tail and the Himalayan have very different calls. So you can easily distinguish across the two species with calls. But within the Himalayan, we didn't find much variability. We haven't done any acoustic analysis for marmots. We have for palm squirrels. And even for one species of palm squirrels, we find high variability. So it's possible that we'll find similar patterns for other species like marmots. For instance, with palm squirrels, some of the things we find are squirrels in Chennai don't sound like squirrels in Kodaikanal. It's the same species, but the frequency is very different.
Okay.
The kind of call is the same, but several acoustic properties of the calls are different.
Just one very silly question, one more last question. So I came across, like I have been to some parts of USA where I saw that the squirrels are really huge and Indian
Yeah.
squirrels are really small, even if in northern India. So I thought that maybe it's because of the weather they are accumulating more fatty tissue, but even in northern parts of India where their temperature is not so much, we have small squirrels. Can you comment to why this will be like? Is this so much difference? I like those are like huge, like rabbits, like I have seen.
So I think you're talking about fox squirrels in the US, which can be really big. They have gray squirrels also much bigger than our squirrels.
Yes, gray squirrel, yeah.
Yeah. Although fox squirrels are arboreal and not in all parts of the US, but they're much bigger than the grays. So India has a high diversity of squirrels. And if we look at all of the squirrels we have in India, actually the largest is the marmot. But we have giant squirrels in forests that are about 2-3 kilos.
And we do have this.
Okay. In India, in many parts of India. So the palm squirrels are one of the smallest squirrels. So yes.
So we do have big squirrels. Okay, good to know that. Okay, thank you. Lovely talk. Thanks a lot.
Thank you.
We have time for a few more questions if there are any questions. John?
Can you Unmute it. Can you hear me?
Yeah.
Hi, Nandini. Good to see you
Hi,
again.
John. Good to see you.
Quick question. You had mentioned the US and Asia for this particular grouping. Did you mean Eurasia?
No, Europe is not, you mean pikas, right?
Yeah.
So Europe is not a place with high pika diversity. So Central Asia is where you have the highest diversity of pikas, from the Himalaya upward into, all of these like 12, 13 countries in Central India. I'm sorry, Central Asia, where you have these extensive mountain ranges and these high plateaus. That's where pika diversity peaks. There's only one species of pika in North America. So they're almost all Asian.
All Asian, but there's one. I've seen them in, I think, Colorado. This is
Right.
But what explains that radiation? I mean, it's odd in some sense, because I would have thought it was just holoctic in one
Yeah.
sense. And
Yeah.
I was trying to figure that out. So what do you think explains it?
I think it is cold adaptation. I think it's not necessarily holactic. And again, I think the kind of cold is almost like a dry hypoxic cold. In most places, pikas seem to be associated with low oxygen. And so many people have studied adaptations to hypoxia in pikas and adaptations to cold. So it's probably not that they're just in cold places or northern latitudes. Even like in California, I think pikas are found on mountaintops. California can get quite hot, and it's not especially known for its cold weather. But pikas are really high up in the mountains. So I think it's a habitat link thing. I'm not a biogeographer, but I think the radiation is across the Bering Strait. So older phylogenies seem to imply that the American pika is probably the ancestor of pikas. Newer studies seem to suggest that the Tibetan plateau is where pikas have evolved and radiated out of. There's an incredible study with a ton of genes. Yeah, that was just out a few years ago. And that seems to imply that the Tibetan plateau is where they evolved from and radiated outward toward. But if you look at fossils of pikas, they are found in Africa and many parts of the world where pikas don't occur today. So I shouldn't be talking about biogeography of pikas.
Another quick question about this, and this is, you said the 5-striped and the 3-striped overlap, but then they move in different directions, right? One towards the north, another towards the south, but they have. Is it similar to the rhesus macaque and the bonnet macaque?
Yeah, so I mean, I was in Aninda Sinha's lab for a few years, and of course, there are strong parallels between the bonnet rhesus line, and so yes, that's something we thought about and said, oh, maybe it applies to squirrels. We don't find too many places where squirrels overlap. We are building a database through citizen science as well as primary field work. And we don't find too many sites where they are in true sympatry. So we are trying to see, one of my PhD students is going to study the differences for, when they occur separately and when they occur together. So you can find Pamaram like close to Tirupati, and Penanti like 2 kilometers away, but you don't see them exactly in the same place. So yeah, we have found a few sites like that, but not too many. But we don't know why or what's going on or how they partition resources or microhabitats.
Okay. I think there's one thing. I'm going to ask one last question, and I'm going to ask my class right now because I'm going to charge back to class. But are there any questions? Okay, someone's just articulating.
Sorry, there is one question in the chat box.
If Ananya, I think she could ask the question herself, if that's fine.
Yeah, I was curious about. Oh, she's right there.
Yes, I was curious about like how you would attribute, like what the indicators were, how you would attribute characteristics to ferret vision and raptor vision, just the differences between them.
Yeah, I think these models are built based on studies of vision systems of animals, and looking at the wavelengths of light that they can see. So there's long wave, medium wave, short wave, and UV. I think these are the 4 channels that any animal can see. Maybe there's a 5th that I don't seem to remember. But different species are known to be able to see different channels of light. I know that some of this work can come with genomics. You know whether an animal is a trichromat or a dichromat from looking at the genome. But some of these are more traditional studies with physiology. I'm not really sure how, but we pick species for which models are available, and we use a proxy for a group. So we use a ferret and extrapolate it to cats, though they're not extremely closely related.
Thank you.
1 second, there's a question here. Go ahead. Mute, okay, fine. Go ahead. 1 second.
Am I audible?
Yeah.
I was just curious to know about the species that you use or the specimens that you used in the museum. And since they were, I'm guessing that they were dated back in time, how do you ensure that they have the same characteristics that you were looking for in your current study?
Yeah, that's a great question. You're asking, how do you know if they have the same characteristics in the wild today, in the same place that they were sampled? Or are you asking a question about?
It was a little hard to hear. Maybe if we could run.
Hi, no, my question was the former that you mentioned. How do you ensure that it has the same characteristics as you would find in the current population of squirrels?
Yeah, that's a good question. Many of these collections are 50 to 80 years old. Some are up to 100 years old. We assume, maybe naively, that this window of time is not enough for color to have changed drastically, unless there's very severe drift or hybridization or some very strong selective force that's causing color to change. If it is simply color that has evolved in response to environmental variables to provide camouflage advantages, we assume that this won't change so rapidly. So it's an assumption, but it's a very valid question. Another aspect to think about when you study museum skins is that color fades over time. So we looked at a lot of studies to see if that is something we should factor in, and we did not find a correlate with age of the specimen. So we went ahead and used all our skins.
Nandini, so go ahead. Quick question again. We spoke
Sure.
about before, and it was nice to see the elements about Sangam literature that you mentioned. But
I'm sorry, there's so little history in that
in
topic.
terms of, say, burrowing behavior and the like, when you do your literature surveys, how far back do they go?
Oh, with the Ladakh system, it's very hard to find.
Can't hear you.
With the Ladakh system, it's very hard to find mention of animals. Sorry, can you hear me now?
Yes. Good.
Yeah. So with the Ladakh system, it's extremely hard to find a mention of either mammoths or pikas in the literature. First, there's very little literature from the region. You can find some British records from a couple of 100 years ago. British explorers have walked some of these mountains. Well, the thing is, people did not pay much attention to these animals. Mammoths are like pests; they're everywhere. So who wants to, record a mammoth when you can record a snow leopard, right? So yes, I think they've been there. And when you talk to Ladakhis, they have stories of mammoths in their culture. We've heard, so I looked at Buddhist literature because there was extensive documentation in these regions. Very little mention of something like the marmot or the pika. So one species of marmot, the long-tailed marmot, in some places burrows in soil where there is gold dust. And so it when it burrows, there's some gold dust that gets thrown up, including on the marmot. And this is along one of the tributaries of the Indus or something. And they call it the golden marmot. So there are some stories about this species. So very, very few though. So it's not something that is iconic or has captured the imagination. And pikas is no mention at all, at least not that I found. So.
Well, no, that's
So Lippy's got a question. Lippy, please, would you like to go ahead?
This question is from something that you mentioned in your slides, in one of your first slides, where you ask, are traits adaptations or are they just evolutionary relics? So how do we differentiate that, and how do we not fall into the trap of teleological thinking? Is there
Yeah. Yeah, that's great. Yeah, it's tough. I think it's easy to think that everything has evolved for a reason, and you break it down into every body part and say there is a reason for everything, and that it has a function today or provides some kind of advantage today. But I think one of the main ways we do this is by looking at comparative phylogenetic analysis. And if you can build a robust relationship of your species of interest and include enough outgroups so that you're kind of not introducing bias, you can answer these questions. And this is a field of evolutionary biology that's just grown over the last 20 years, 30 years. It's grown exponentially. So, and with genetics, we have better and better phylogenies, so we can use these tools to answer questions regarding evolution, which is quite cool.
Okay. Well, thank you so much, Nandini. This was an excellent talk, and I loved all the questions that came from among faculty members and students. And I wish, and I'm sure we all wish, that when down the line you can actually physically come to campus, and since you're so close, and interact with all of us. And with that, we'd like to end this year's BIOS seminar series. And thank you so much.
Thank you so much for having me. It was really lovely to interact with all of you last weekend, and yeah, I'm looking forward to visiting campus.
Thank you.
Thank you.
Thank you, Nandini. Bye.
Bye.